takes a few seconds. Sorry. Okay, well, I think it's exactly 6 and right on dot. I welcome everyone who is following us through the YouTube channel, the TIFR Platinum Jubilee channel, as well as those who have been kind enough to join in Zoom as an audience to this edition of the NSF Wednesday Colloquium. This is the first of the year, and we actually are very blessed that Professor Yamuna Krishnan is actually helping us start the series this year in 2022. So I wish all of you a happy new year from my side. And I am sure whatever challenges we are going to face this year, we are going to sort of figure out ways to battle it out. So let's start with a positive note, even though we know the third wave is right on. So let's keep ourselves safe. I just wanted to start the series again for those people who are joining us in the audience for the first time, those students, scholars who are joining for the first time. The NSF Wednesday Colloquium actually is a very old series within the TIFR tradition. It was started and envisioned by our founding director, Professor Homi Bhabha, who thought that science doesn't require a subject-specific flavor, and it is actually done in totality. Because of that idea, the Institute, of course, started with having physicists, chemists, and biologists all together under one roof. And this particular session on Wednesdays at 4 p.m., that is the traditional time, at least all the scientists from around the Institute gathered together once a week and listened to eminent experts traveling to TIFR for giving a talk. And at that time, everybody got together and listened to talks in physics, chemistry, and biology with the same even keel, and enabled collaborative ideas within the institute as well as with the person who is actually giving the talk. So that really strengthened the kind of science that is going on and the kind of thought that students were getting shaped into. So I'm very proud to be part of this series and hosting this. We have an eminent speaker today, one of our own, actually, I might say, who actually was a faculty in a TIFR institute at NCBS in Bangalore for many years, from 2008 to 2009. And Professor Yamuna Krishnan, who is currently with University of Chicago, our own loss, but she has gladly agreed for this colloquium. So I will not waste much time, and I will give my colleague, Professor Sandhya Koshika, a chance to introduce her. Sandhya has been a long-standing friend and collaborator. So Sandhya, please.
Thank you, Jyotishma. Thank you very much, Yamuna, for agreeing to speak here. And I'm really looking forward to your talk. Yamuna is an unusual scientist. I have known her for several years now. I first started thinking of her as an inventor because she would make these really, really cool devices which moved, sensed, and did all kinds of wonderful things in cells. And using her inventions, she has been discovering new biology. And one of her most recent papers is phenomenal. It sort of tells you very beautiful things about lysosome biology. And I have seen, and in fact, the last talk she gave at TIFR was the Infosys Prize talk, sort of related to the Infosys Prize talk, where they encourage their winners to speak in public fora, where she solved a 30-year-old problem using her sensor in discovering molecules that moved ions across the membrane, across the lysosomal membrane or endosomal membrane. So she is an inventor who has also become a discoverer. And during this journey, she has learned more biology than I've ever seen any non-biologist do in my limited view of the world. And it has been really fun to watch her research program. She's a terrific mentor. A lot of her former postdocs and students are now part of the Indian ecosystem. And it's with great eagerness I look forward to her talk today. Thank you, Yamuna.
Thank you, Sandhya, for such a wonderful introduction. So I also want to thank Jyotishman, for this generous invitation to deliver this Institute colloquium, this NSF colloquium. Jyotishman asked me to talk about our most recent work on how DNA nanodevices attenuate tumors and its potential applications in medicine. And if I also count my first 2 job talks here when I was a postdoc in 2004, I was just counting, this would probably be my 6th or maybe 7th lecture at TIFR over 17 years. It's always been my toughest audience because, well, when you're with your family, they know you. And so there's no point trying to convince them that you're doing anything useful. And so with the times being what they are, and basic science coming under threat in every corner of the world, I thought that I would try and shine a light on the role of fundamental research underlying what now seems to be potentially useful development from my lab. And so when I joined your satellite ship in NCBS in 2005, I wanted to know the answer to a fundamental question related to DNA structure that had no utility to either myself or anyone else. And to this day, I don't know why Vijay hired me. Anyway, this esoteric question gradually morphed into a DNA-based technology that now has the potential to make certain untreatable cancers treatable. So I want to detail this metamorphosis. And some of you may have seen, detailed snapshots at different time points when I've given talks at TIFR, where I talked just about one particular finding. But I thought today I'll try and stitch it all together and try to feel out the arc that sort of connects blue sky exploration to something useful, or at least useful to a few mice that had cancer. Okay, so this question, blue sky question that I had was about a 4-stranded structure that was made from a particular nucleobase called cytosine, where many cytosines together would form this kind of a 4-stranded structure. And my question was, why is this structure 4-stranded? Because DNA is conventionally 2-stranded. Now, the structure was 4-stranded also because it would form only under certain acidic conditions. And I wondered whether we could use this to make a pH sensor out of nucleic acids, where you could take an assembly and use it to report on solution acidity. Having done that, we then asked, can it do the same thing, but inside a compartment within a living cell called an organelle. So organelles are small compartments within cells, and just like organs perform functions for the body, organelles perform various functions for cells, and they are at very, very different pHs. So I just wondered whether it could perform a pH reporting function inside an organelle. Having done that, we then asked, can it do so inside a live cell that is part of a whole living animal? And then at each point, the question led to an answer, which was like, yes. And so we then jumped to the next interesting question to us, which was, if it could sense pH in a specific organelle inside a specific cell, could we reroute it and send a DNA nanodevice to a different organelle in a different cell of a live animal? And lastly, since we were sensing pH in all of these locations, we asked, can we sense other ions, other chemicals inside cells? And can we chemically map out organelles? Because these are unknown spaces inside the cell where we actually don't have an idea of what their chemical composition is. So from a basic point of view, can we just find out what the environment is? And finally, we recently started asking, what is the consequence of these chemical maps? Yes, you can tell the chemical content, but does it actually reflect organelle state and organelle function? And the recent work where it started actually showing that it could probably be useful was that we said that, well, now that we can send DNA devices to any specific compartment within any specific cell, can we change the chemistry there? And can we modulate organelle state? By modulating organelle state, can you flip cell state? And by flipping cell state, can you then change the biology of an organism? If you can flip the lysosome in a given cell, change the cell state, can this altered cell state now correct disease in a whole organism? And so I want to take you through each of these steps, and I will try to convey that had any one of these steps been missing, and importantly, the very first step, if that had been missing and we had not asked this very simple question, we would not be where we are today. So in 2005, when I joined NCBS, I was fascinated by a field called structural DNA nanotechnology. Its founder, Ned Seaman, started it when he constructed a nanoscale cube from 10 strands of DNA. And he found that when matching strands paired with each other, giving you this iconic DNA duplex, the resultant duplexes, or the double helices, stiffened up these DNA filaments into rods, forming an assembly that had the connectivity of a cube. But it was not very efficient. The yields were about 1 to 3%, which is, if I were to take 100 such strands, maybe I would get 1 cube out of it. The next leap came when Andrew Turberfield at Oxford made a tetrahedron, but with 98% yield. Now we were approaching high precision architectures based on DNA, where we might actually achieve some practical applications since they could be made efficiently. So in parallel, there was a different class of precision architectures that was emerging. And here, these were called dynamic architectures that could move. Small modules of DNA, whether they were little rods or little triangles like so, could be made to undergo specific movements, switching from one specific conformation into another. And here the movement is powered by the addition of DNA strands, which upon binding the architecture either pinched it or relaxed it. And other ways were by even changing the twist of the DNA helix. And so one could even use the space enclosed by DNA helices to encapsulate little cargo and then release it by changing the dimensions of the surrounding polyhedron that it was trapped in. So you can see that complex DNA systems can be used to display other functional materials and leverage their signature capabilities. So in general, if a material can be chemically conjugated to DNA, then a DNA structure can both inherit the properties of that material as well as offer control over organizing that material on the nanoscale to enhance the material's performance. So in 2005, when I started my lab, I was thinking about this unusual 4-stranded DNA structure that is formed from DNA strands that are comprised of only one nucleobase. The DNA has 4 nucleobases: adenine, thymine, guanine, cytosine. And this structure had only cytosines in it. 2 cytosines would pick up a proton between them and base pair to give you what is called a C+ base pair. And you would have a ring of such C+ base pairs on 2 strands that would dimerize into a duplex. But it didn't stop there. 2 such duplexes would intercalate to give you a tetraplex. And my little question at that time was, I wanted to know why the structure was 4-stranded, given that in a dimer, all the bases were fully paired. And so there's no necessity to apparently throw entropy out of the window and further dimerize, right? And this was when Shovik joined me. And the study helped us both become familiar with the dimensions of this unusual structure called an I motif. And we found that it would form only under acidic conditions. So I wondered whether we could use it to make a pH sensor. So my first PhD student, Shovik, and I, we designed an assembly that had a cytosine-rich strand here that would fold into an I motif, into this four-stranded structure at acidic conditions. But at neutral pH, it was designed so that it would exist in an extended state. We stuck two fluorescent dyes, one red, one green, and these acted as what we called a fret pair. In a sense, when the distance between these two dyes are small, you have low green fluorescence and high red fluorescence. And when they are far apart like so, it's the opposite. And so if you now were to take a ratio of green fluorescence to red fluorescence, we found that it was a beautiful reporter of solution pH. And so the environment of NCBS was then responsible for the next direction that was unpredictable. So there was a strong school of endocytosis around, given Jitu's lab and many other labs. And I became aware from the students there that, all endosomes, all compartments inside the cell were not alike. I used to think of all endosomes as being similar. What I learned was that an endosome starts out life as an early endosome and then undergoes a series of maturation steps to finally becoming a lysosome. Now, what is an endosome? A cell, just like us, has to feed itself. And so what it does is it engulfs material from outside and it forms a little fat bubble in the cell containing its food. Just like us when we eat food, right? So these fat bubbles are called endosomes, and they undergo a characteristic series of maturation steps, finally ending up in an organelle called the lysosome that is generally referred to as the stomach of the cell, where digestion of the cargo happens. So all along endosomal maturation, where it goes from an early endosome to a late endosome to a lysosome, the pH inside the organelle becomes more and more acidic. So I then wondered whether I could use my pH sensor to follow this process. Fortunately, I could persuade Shovik to try it. He was skeptical. But once the moment he saw the first results, he was off to the races. And then after that, I was basically following, and he taught me everything that I would later need to know. So in a nutshell, he found that if he added a DNA device to a living cell, it would bind a protein on the cell surface called a scavenger receptor that would attract it. And this would drag the DNA device into an early endosome that then matured into a late endosome and finally a lysosome. So let me tell you how we make these pH maps and how we are able to visualize this. So here's an image of some endosomes in a cell that contain some of our DNA nanodevices. Using a fluorescence microscope, you can take an image of the green fluorophore, and this is what you see in this green channel, in this green image, and similarly one of the red fluorophore. Then you can compute the ratio of green intensity over red intensity in both images at every pixel. And this gives you a heat map of the cell. So wherever you see blue, it's highly acidic pH. Wherever you see red, it's more neutral pH. And so using our pH reporting DNA nano device, we mapped out how endosomal pH changed along the entire process of maturation of an endosome. Okay, the surprise in all of this was that the performance of the DNA nano device inside a glass cuvette shown in red and that of a living cell shown in blue were nearly identical. And this meant that our devices were not interfering with, nor were they being interfered by the cell's own networks of DNA control. So this device was actually behaving like a true passive reporter inside the cell. And so Sandhya, Koshika, and I, meanwhile, had struck up a friendship at NCBS. And one look at worms under the microscope in her lab, I fell in love with that organism. And I wondered whether these devices could function inside a live cell that was inside a whole animal, instead of taking cells inside a petri dish and following endosomal maturation over there. And Sunaina Surana, who was a geneticist, who joined me exactly at that time when I was thinking about this possibility, said that she was interested in exploring this question. And we both went to Sandhya, and she didn't need any convincing. And so together we showed that if you injected these DNA devices into worms, they went and labeled only one particular cell type. There are 6 cells like this inside C. elegans, and they are called coelomocytes. And they went and labeled these cells, and you can see here the coelomocytes are labeled in green, and our DNA device is labeled in red. And these are some endosomes inside coelomocytes. And so you can see that wherever you have coelomocyte lysosomes, our DNA device has gone over there. And so we found that DNA devices went to this particular location only because these cells had loads of scavenger receptors on them. This is the same receptor that was present on the cells where DNA went into endosomes in cell culture. And inside a whole animal, you had these cells expressing this particular receptor, and DNA devices were attracted to these cells. And the other interesting thing is, in these particular cells, endocytosis is on overdrive. They are very, very hungry cells. And so naturally, they mopped up all the DNA devices. And so here you see 2 coelomocytes where the endosomes have green fluorescence. And at the time, I was excited that these DNA devices went exclusively to one set of cells along one pathway, labeling a particular endosomes on one particular pathway. But I didn't really start to appreciate how important this would be until much later. Anyway, what we went on to do was to show that you could indeed map out endosomal maturation inside a live organism. And we didn't realize that we were the first to do that at the time. But what struck me at that time when I saw this was that device performance inside a live cell, inside a live organism, was as high as device performance inside a glass cuvette. This again meant that you had something that was autonomous, independent of the complexity of the milieu that it was in. It was just doing the one function that it was engineered to do, not interfering with the complex system, nor letting the complex system interfere with its own function. Okay, so then it became clear that if DNA devices found the right receptor, they would end up in a specific target organelle. Like if it bound the scavenger receptor, it would end up in the lysosome. Shovik then showed that if he made a DNA device bind a protein on the cell surface that was destined for another organelle, he could now reroute devices to end up in completely different organelles, like the Golgi, which is called the post office of the cell, or the recycling endosome. All he needed to do was to find a protein that could act like a shuttle between the cell surface and the organelle. It had to have high enough abundance on the target organelle and just enough abundance on the cell surface. So if he attached, say, a molecular motif on the DNA device, that would enable it to latch onto the part of the shuttle protein that was exposed to the outside of the cell, then the shuttling process would enrich the DNA device in the target organelle. It's just like us getting on a bus to go to another location. If you have the right ticket, you will be able to get onto the bus that will take you to your destination of choice. And so we were able to show that you could indeed reroute DNA devices by latching onto a specific shuttle protein at the plasma membrane. So if you recall, when we injected DNA devices into worms, it ended up in lysosomes of coelomocytes because that was the default pathway. When Shovik found that DNA nanodevices could be rerouted to other organelles inside simple living cells, Sandhya and I wondered if we could now reroute them to label her favorite cells, which are neurons instead. And at the time, this was 2012 to 2015, our attempts didn't work, mainly because I think the genetic tricks at that time weren't as sophisticated. But then we revisited this a few years later, around 2017 or so, when genetic tricks became much more sophisticated, along with Paschalis Kratsios, another C. elegans geneticist who was based at UChicago, who I just came across, and he was excited to try this. But the idea itself did not change at all. The strategy of labeling didn't change at all. Everything, it was just the genetic trick that was slightly different. So in a sense, the idea that was essentially unchanged that worked was developed by Sandhya and me, both of us screaming at each other in excitement in our small offices in NCBS. We fused, our idea was to fuse a DNA binding protein. Let's call this DNA binding protein 9E. We fused it to a cell surface receptor, a protein that's on the cell surface. And we used a genetic technique to make worms express this protein only on the plasma membrane of neurons and no other cell type. Then we found that if you injected nano devices into the pseudocoelom of such worms, they exclusively went and labeled only the neuronal membrane surface and no other cellular surface. So clearly DNA devices could be attracted to wherever it found a receptor that could bind it tightly. So it was clear we have cellular level specificity, but can you get now organelle level specificity? On top of the cell that you're going to, can you get to a specific organelle in that cell? So what we did was we fused this 9E to a protein called synaptobrevin. And synaptobrevin has this shuttling characteristic. Some of it is present on the plasma membrane, large amount of it is present in endosomes. So we wondered, it should now be able to take DNA devices from outside and put them in endosomes inside neurons. And so when we did this and expressed this fusion protein of 9E, we fused 9E to synaptobrevin. And now synaptobrevin is carrying this receptor for DNA on its back. And we use these genetic tricks to make sure that we could express this protein in, you can choose now which neuron you want to express your protein in. They could be cholinergic neurons, they could be glutamatergic neurons. Subsets of neurons can now be made to express whatever protein you want. And Paschalis had developed a lot of this technology himself. And so what we did was when we took worms expressing this protein in specific neurons, we found that if you inject nano devices into these worms, they specifically label only lysosomes in whichever set of neurons is expressing our protein. So here we are using an artificial synthetic DNA binding protein that we developed called 9E. So we then wondered whether you can achieve such tissue-specific and organelle-specific labeling even without these genetic tricks, provided you could find the right receptor. So, for example, the intestine of C. elegans comprises gut cells called intestinal epithelial cells, and they express a protein called SID2 that binds double-stranded RNA. And the reason that RNAi or RNA interference works so well is that this particular organism expresses a lot of this RNA binding protein called SID2. So what we did was we attached a ligand for SID2, which was double-stranded RNA, to our DNA device. So now our DNA device has a motif that SID2 can see and like and can grab onto. And so when we did this and attached such a DNA nanodevice and made worms drink our DNA nanodevice such that it now filled the gut, this nanodevice nicely labeled lysosomes in this particular cell type. And so I was now quite excited that we were able to send DNA nanodevices to a specific organelle in a cell type of our choosing. So I spent quite a bit of time looking for molecular systems that can achieve both tissue specificity as well as organelle specificity when they are extraneously introduced into a whole multicellular organism. And I failed to find any, but I found a few that do so with tissue level precision. And I include some of these here: CRISPR, RNA interference, click chemistry. The only one that I found which has tissue level and organelle level specificity is GFP technology. It can achieve this precision, but GFP has to be genetically encoded. The only thing that can do so where you externally introduce it into an organism that doesn't have genetic encoding are naturally occurring viruses or synthetic viruses that have been modified. These can achieve both cellular and subcellular level specificity. So now this places DNA nanodevices amongst a vanishingly small number of molecular scaffolds with which one can achieve high precision biological targeting. And I believe such high precision tools might now be ready for biological manipulation that could empower the next generation of organelle targeted therapeutics. And I'll get to that in just a little bit, but allow me to take you on this arc. So on the one hand is where you can measure, which is what I've been talking about. On the other hand is what can you measure? And as all of this was going on, this was a 10-year saga trying to send DNA devices into different organelles of whole organisms. And so as all of this was going on, we were wondering whether we could map chemicals other than protons inside organelles. You see, as a chemist, I knew that other chemists like Chris Chang at UC Berkeley and Nagano in Japan had discovered small molecules that could fluoresce upon binding specific ions or reactive species such as superoxide or nitric oxide. These are all important molecules for biologists that they've always wanted to see, but they've been very limited as to looking at how they fluctuate inside living systems. But if you add these small probes to cells, they are very small, so they'll diffuse all over the cell. And so they paint the whole cell. And so you cannot get location-specific information on how much of your chemical is there and where it is. With green fluorescent protein, one which is used by biologists a lot, you can mark exactly your organelle of interest, but you're very limited in terms of knowing the concentration of chemicals over there. All you can see is, here is your organelle. Maybe you can tell pH or possibly calcium, but you can't tell anything else. With DNA, we realized that you can localize your detector probe, which is your small molecule, to a specific organelle, like I showed you. And because DNA duplexes are always in a 1:1 stoichiometry, you can attach another dye as an unchanging reference signal in a different color. So now the ratio between detector and reference turns this qualitative high-low kind of signal into a quantitative measure. And you can tell how many millimolar or micromolar of your chemical is present in the environment surrounding your DNA device. Importantly, a DNA device is too huge to be able to smash through or diffuse through the membrane of an organelle in which it is finally located. So it's trapped. So given the stable spatial localization, confinement of your DNA inside an organelle, you should be able to measure and get chemical maps inside your organelle. Then there was a burst of sensors surrounding this concept, showing that this was in fact possible. And it happened only because there was a group of highly creative, almost joyously crazy young people who all joined at the same time, and then they fed off each other's enthusiasm. So Sonali and Kasturi showed us that we could use a chloride-sensitive dye to map chloride. Maulik, Junni, and Anish, who is now at TIFR Hyderabad, showed that you could map a reactive species like reactive oxygen species or reactive nitrogen species, which biologists have always wanted to see and could not see in specific organelles of cells. Anand showed that you could measure membrane potential in organelles. And Kaho and Krishna showed that you could quantitate enzyme cleavage inside organelles using DNA nano devices as reporters. So I'll discuss one example, and I'll talk about Kasturi's sensor for calcium, just to get across how we can do this for one ion, and then you can just see how it works for all the rest. Where Kasturi showed that you could measure calcium inside lysosomes. So for 40 years, biologists have always wanted to measure calcium inside lysosomes, except they couldn't because the lysosome is highly acidic. And why is this? Because every calcium sensor that we know fails under acidic conditions because they all work by binding calcium through carboxylate groups that get protonated when the acidity is high. So basically, you cannot use a calcium sensor like this to map calcium in lysosomes because the dyes also cross-react with and sense acidic pH. So we turned the problem on its head. If you take a duplex with a 1:1 ratio of a calcium sensing dye that glows, say, orange, and a reference dye that glows, say, red, then the ratio of orange to red will be dependent only on two things, right? pH and calcium. The problem that had biologists running in the opposite direction was that you will never be able to tell how much came from pH and how much came from calcium to this orange fluorescence. But we had a pH sensor. So if you could append a pH sensor onto this system, then that at that very same location, you can measure pH. So if you know exactly how this orange dye's calcium sensing characteristics change as a function of pH, then you can deconvolute and get the precise contribution of calcium to this orange signal that you have. This was the basic idea. So Nagarjun and Kasturi made a DNA nano device that carries a calcium sensitive dye called ROD5F that glows orange, and Alexa 647 as a dye that glows red, and got these orange to red ratios as a function of increasing calcium and at every pH. And so you could map out this exact how it would, what kind of signal you would get at every single value of calcium and pH. And so from this, you could determine how the affinity of this calcium sensing dye changed as a function of pH. And that is, you can plot this affinity as a function of pH. And so now, here are the first maps of acidic calcium stores, acidic calcium organelles, basically lysosomes inside cells. And here's how Anand obtained them. I told you how we get a D by A map, a green to red map. And you know exactly how this value of green to red depends on pH. There's an equation that can describe this. So you can convert this into a pH map. You know exactly how the calcium sensing dye's affinity changes as a function of pH. There's an equation that describes this. So you can convert this pH map into what I call a KD map for dissociation, or the affinity for this calcium sensitive dye. And you know how the calcium sensitive dye has reacted at that pH. So you can introduce this as a pH correction factor and then get the map of absolute calcium. And so the reason why I'm taking some time here to show you this is because these are the first quantitative calcium maps in any organelle. They happen to be in a living organism like C. elegans, which is also a statement as to how bright these dyes are. Usually chemists will first do it inside live cells and then show it inside a living organism. We just went straight to a living organism. So the ROD5 family dyes, these calcium sensitive dyes, were known for 30 years. pH sensing dyes have been known for even longer. The fact that you can have DNA or endocytic tracers to mark organelle lumens was known for 50 years. And we've all known for 40 years that we've always wanted to measure lysosomal calcium. But despite having all these working parts, right, these maps could only be visualized by bringing a DNA duplex into the equation, because DNA gives you a stoichiometric precision with which you can display a ROD5 dye, a pH sensing dye on a scaffold that is that can act like an endocytic tracer. And it was only getting all these into the mix that empowered us to get sort of calcium maps inside the cell. So we then asked, you know, having measured a whole range of chemicals that people have always wanted to measure inside cells, we asked whether these chemical maps had any connection to the state of the organelle or its function. And if it did, then the corollary is that if you exclusively modulate the state of the organelle, can you change its function and thereby affect the function of the cell as a whole?
Yamuna?
So here's, yes.
Sorry, interrupting you. Can you go back?
Yeah. Yes.
So I just wanted to know, yes, right here. So you have constructed a KD map.
Yes.
And just wanted to know what is the, the concentration itself is really unknown, right? Or you had some idea or estimate of the concentration of the calcium. And the other thing is, if you were to find the KD map now, you can actually target it better, right? So in some sense, what I'm trying to say is the equilibrium of these sensors with the, how well do we understand that?
Very nice. So the first thing that we did, right, the very first dye that we made, we first had to see what, we have to be lucky that your dye has a KD value that is compatible with the physiological concentration of calcium, which you don't know. So you are really groping in the dark, right? But fortunately for calcium, people gave us an order of magnitude value. So calcium outside the cell is in millimolar range, 1 to 5 millimolar. Calcium in the cytoplasm, that is somewhere here, is in the nanomolar range, 100 nanomolar. Calcium in the ER is also micromolar, just like extracellular. But calcium in the lysosome is somewhere in the middle. So they said we think it's micromolar. Some prior crude estimates suggested hundreds of micromolar. The first calcium dye that we used was fully saturated. So we then said, okay, we need to make it weaker, right? So this rod 5F is a new calcium dye that Nagarjun had to synthesize to bring down its affinity for calcium to the right value. And then we did this. So what took 3 years could actually have been done in 1.5. But that was because we didn't know what value we were supposed to be seeing. So yes, that's
okay, that's great. Thank you. The other thing is, what is the typical sort of kinetics of these? When you see something that glows up, or so my question would be: there is a time scale for taking an image, and there is a time scale for fluctuations in the calcium concentration as an event is happening, a biological event is happening.
Yes.
So what is the temporal sensitivity
resolution
of
yes. So these if you're looking at the concentration of chemicals and the concentration of our nano devices, our dyes in the solution, and we've done these calculations, we've done these estimations of experimental estimations. We have roughly about 200 to 500 devices per organelle. So with these concentrations, the size of the organelle, sensing actually is on the high microsecond time scale, high nanoseconds time scale. Response of the dye itself is in nanoseconds, right? So, so many of these dyes. So the question then is, you can actually image in millisecond time scales. So your dye is already fully responded. Your systems have already responded. You're limited by imaging speed.
Okay.
The second is, what are the time scales in which biology works, right?
Right.
Could you have changes in microsecond time scales that are important? We don't know this, right? Because biologists have so far been looking at things which are can be looked at millisecond time scales, because that's the speed of imaging. If we can go faster, we might be able to see something. But at the moment, your probes are not limiting. It is
the probes are not limiting. It's essentially
probes are not limiting. It's the optics and imaging that would be limiting.
Okay, so you just need a little bit more time to collect the images to good signal to noise. That's what you presume.
Yes.
Okay.
Yes.
Okay. Now the other thing is, if indeed that is very true, then you will smudge out all the fluctuations in the microsecond and only see the heat maps in milliseconds as they are.
I'm coming to that. That's going to be the next next part of what I'm going to talk about.
Thank you very much.
So that's a great question, and thank you for sort of kickstarting this part. So we were then asking, right, whether you can make chemical maps of organelles and whether these chemical maps connected to the state of the organelle and could potentially affect organelle function. And if it did, of course, then it would be great if you could just specifically modulate organelle function or organelle state, and then see if you can affect the whole cell state. So the system we are going to look at is the lysosome. And there are two different states of the lysosome. Lysosomes are normally vesicular, but sometimes in immune cells, if your cells get inflamed, right, or they get activated, immune cells get activated, or if your cells are starving, right, then the lysosomes can sometimes become highly tubulated. And so there are these two states. And the immune cells we're going to look at are called macrophages. And this roughly translates into big eater. And the functional part of a big eater is its stomach. And the lysosomes are generally thought of as the stomach of the cell, as I mentioned earlier. So they digest the nutrients that the cell eats and then sends it out. The digested material, all your amino acids and sugars and everything are sent out to the rest of the cell. So, but give me a minute to tell you what is it we are attempting to test and take you through my thought process. See, biologists have already shown that cell state is connected to cell function. Take, for example, an immune cell in its resting state, right? If it is starving, right, it amplifies a process called autophagy, self-eating. That is, the lysosome is cannibalizing older parts of the cell from within in an effort to provide the starving cell with building blocks that it needs to make new proteins. And this requires the cell to adopt a specific transcription program. So the cell now adopts a state that is a different state from the resting state. On the other hand, if the cell encounters a pathogen, it activates a process called inflammation. This sets off a different transcription program for immune activation, and the cell adopts the inflamed or the activated state. And so we know that organelles in the cell, specifically the lysosome, they rise to meet each kind of challenge. And they do this by changing their function to help drive the cell's new overall function. So during autophagy or inflammation, it is the lysosome in immune cells that senses that, oh, there's no food coming, I'm starving. Or, oh, I've just eaten a pathogen, and sends these signals to the nucleus to initiate the signaling. So we know that, I mean, biologists have shown that the nature of the lysosome changes in each of these cell states. Now, what we are trying to ask is the reverse. We know that there must be some feedback, right, between the nucleus and the lysosome, because once the pathogen is cleared or once nutrients are available, the lysosome has to communicate that state to the nucleus and switch the nucleus and cell back to the resting state and reset the transcription program. And so first, we want to test whether we can explicitly change organelle state, like say the morphology, and ask, does it change the chemical and biochemical properties of the organelle? And if so, can we change, if this is the case, and we can change organelle state, can we, if this exists, change cell state or cell behavior? So Bhavya designed a DNA device that bound a family of proteins called the Q proteins that were on the cell surface, because she wanted to transport DNA devices to the endoplasmic reticulum, which is another organelle. But instead, what she found was that when she added it to macrophages, it didn't go to the ER as she wanted, but instead it kickstarted a different signaling pathway that she then delineated that caused very nice spherical lysosomes to now elongate into these tubular forms. Not one to be disheartened, she wondered whether, is there any chemical difference, right, between the lumen of these tubular lysosomes and these vesicular lysosomes? And why does the cell need two different shapes of the same organelle? Is there anything different between them? So she used our calcium sensor, and she used Kasturi's sensor for enzyme activity. And what she found was, all along the tubular lysosome, there was a pH gradient. And in the opposite direction, there was a calcium gradient. So one end of the tubular lysosome had high calcium and low acidity. The other end had high acidity and low calcium. And so this gradient had been predicted theoretically by Professor Botello in Canada. But using our DNA devices, we were able to provide the first experimental proof of this. An even more interesting fact is that when Bhavya used a sensor for cathepsin, which is an enzyme that is responsible for this proteolytic digestion and degradation. What she found was that degradation by enzymes like cathepsin was much higher in spherical lysosomes and much lower in tubular lysosomes. And so initially, finally, at the end of the day, Bhavya found that when she explicitly tubulated her lysosomes, just by adding to this DNA nanodevice that she calls TUDA, if you can just add TUDA and tubulate the lysosomes, this kickstarted this signaling pathway that then led to the secretion of an enzyme called MMP9, matrix metalloprotease 9. And this enhanced secretion changed the nature of the cell and made it much more phagocytic. So a macrophage would become much more bitey or gulpy, or maybe it starts actively engulfing far more amount of material all around it. And so clearly, you could change the nature of the cell by just changing the shape of one of the organelles called the lysosome. So now, at the time that Bhavya and Kasturi were using a nanodevice to measure degradation brought about by cathepsin in the lysosome, a colleague, Lev Becker, on the floor above ours, had been measuring protein content in macrophages. And he is a macrophage biologist. And from him, I learned that macrophages generally occupy two states. One state is a state called M1, and I call these the loudmouths. They take in food, half eat it, then smear it all over the cell surface, and as if to say, look, everyone, look at what I just ate. And this is a great way to tell killer T cells that I just half ate a virus. Here's the remainder. Here's the antigen. Go and find other viruses that look like this antigen and finish the job for me. And he found that M1 macrophages had low cathepsin. Then you have the M2 macrophages, and these have, he found, had high cathepsin. And these guys are different. They are like, shh, they don't want to tell anybody who they ate or what they ate. And this is very important because if you eat something like a blood clot, which is your own, then you don't want anybody to know or get inflamed by your own tissue. So Lev had found, Lev and his student Chang had found that M2 max had high cathepsins. So Kasturi used her DNA-based cathepsin sensor, and Lev is a cancer biologist studying macrophages and cancer. And he found that if you take tumors, 50% of the cell mass in a tumor are actually macrophages called tumor-associated macrophages. And in this context, when he separated M1 and M2, he found that M2s had very high amount of cathepsin. And he just didn't know that it was cathepsin in the lysosome. And he and Kasturi together showed that this high cathepsin activity that he found in M2 macrophages was because of cathepsin that was localized in the lysosome of these macrophages. And so Lev's idea was to try and see if we could make a DNA device that would inhibit cathepsin in lysosomes of tumor-associated macrophages and hopefully try to revert a disease state to a normal state. So let me tell you Lev's reasoning, right? And I told you that 50% of the cells in a tumor are tumor-associated macrophages, otherwise called TAMs. See, you and I are getting cancer every day, but our cells kill these cancerous cells. So when a tumor grows and you have cancer, it basically indicates a failure of your immune system. Your T cells have failed to see the tumor cells. And the reason why this happens according to Lev and many other biologists, is that TAMs in the tumor are this super hungry M2 type. So they gobble up every bit of dead or dying tumor cell, and they never present any antigen to the T cell to call it there, to finish off this tumor. So Lev reasoned that because you have such high cathepsin inside the lysosomes, they are over-digesting, hyper-digesting any antigen. And so if we were to put a DNA device that had cathepsin in the lysosomes of these M2 kinds of macrophages, then they won't over-digest antigens. They will behave like M1s and spit out any remaining undigested antigen onto the cell surface. And the T cells will now be able to see these tumors and then take care of it. And so thankfully, so when he told me about this I told him, like, sorry, I don't think we'll be able to do this. We are sensor people. I actually don't think this is going to work. But credit to Lev, he didn't give up on me. He just went out and got more data to convince us. So what he did was, using a genetic trick, he reduced the number of lysosomes in these macrophages in an effort to reduce the overall degradation by lysosomes. And he did this in 3 different cancers. And he showed that just by reducing degradation in lysosomes in a very non-specific way, you could prevent this tumor growth. And he showed that it prevented tumor growth because these TAMs were basically starting to present antigens and dragging T cells to the tumor. And so by this time, Kasturi, without telling me, was already convinced, and she made a DNA nano device with a cathepsin inhibitor, as well as a tracking fluorophore. And she and Chung, without telling either of us, injected it into mice. And lo and behold, it went straight to only tumor-associated macrophages, didn't mark any other cells in the tumor, selectively to these tumor cells, and no other cell type. And even out there, once it reached there, it was straight in the lysosomes of these tumor-associated macrophages and no other cell type. Now, Kasturi had already known, given her own work in C. elegans and Anisha's work in zebrafish, that if you inject DNA devices into any of these organisms, it will find macrophages in that organism and label it. What I would not have predicted even in a month of Sundays is that this would be preserved even in a much higher organism like a mouse, which is much more complex, far more number of tissues, far more number of biological barriers. We still have to think about why it's going specifically to tumor-associated macrophages and not macrophages elsewhere. But here's the result, and we can't argue with it. And this was also why I was not very convinced when Lev came to me and told me that this might work, because I could see all these other places that the DNA device might end up in. Okay, so the rest went as Lev predicted. The nano devices did indeed inhibit cathepsins in the lysosomes of tumor-associated macrophages. Now, with rocks in their stomach, this uncontrolled tearing to bits of antigens had been transformed into sort of like polite snipping. And antigens are now displayed on the surface of TAMs. And TAMs indeed can now educate the T cells to see the tumor that is present in. And as a result, tumor size reduces. If you combine this with a treatment that causes tumor cells to die, like low-dose cyclophosphamide. If you add this, your tumor cells will die. And so this supplies the TAMs with more amount of tumor antigens. So basically more food to digest and display. Then the tumors are cleared with an efficiency that is extremely exciting for people who have been in this business for decades now. See, many of us are now familiar with the concept of drug targeting, enriching a drug at a tissue of interest, because more drug becomes available at the site where it's needed, and we have less side effects from it hitting places where it's not needed. We know that many cellular processes are controlled at the level of organelles. And now, given our work, you can see that there's a direct line of communication between organelles and cell state. So our work so far has led me to posit that there will be much to be gained from achieving an additional level of drug targeting, where we not only send drugs to specific cell types, but to specific organelles in that cell type. And so I posit that we might all have to start viewing cells the way cell biologists always have, with all of their organelles, if we are to solve the next set of currently intractable problems. I'm going to end by just saying that I've been very lucky to have worked with some incredible students and postdocs, and especially wonderful collaborators, on a rather unconventional direction for our field. And hopefully we've been able to convince you that if even a simple DNA duplex programmed with a few small molecules can do something useful, and that we can learn something useful about biological systems, then the rest of the field that is making these very complex architectures, we will be able to actually achieve much more, will be capable of much more than what we've done. So with this, I will stop and take any questions, because I've been acknowledging people all along the way during the talk, so that we can smoothly transition into questions.
Thank you very much, Yamuna. Thank you very much for this wonderful talk and work that is really opening up new things for everyone. So I would like everyone to just give a warm round of applause. If you can unmute your audios, please, that will give us a feel. Thank you. Thank you, Yamuna. Sorry, I'm a bit persuasive with my audience, so it's okay. Okay. So now we have questions. So please unmute yourself, ask questions. There are 2 questions already on the chat box. I will take them. Let me first take the audio questions. Yeah, Koti.
Hi, Yamuna. Nice to see you and have you here for the talk. If not physically, virtually. Would like to have you physically here sometime. Okay. So I have a couple of questions. One on the work you presented, excellent work. So I have a question on this tubular lysosomes, right?
Yes.
I mean, I'm trying to understand the genesis or the mode of formation, because they almost look linear, not curved or zigzag shape you would expect if they are really long. Is it because they are collapsed from small vesicles of different pH and different calcium that's actually giving the calcium and pH gradient, or is it a different way they are actually forming?
So actually you do see lysosomes which are sort of not completely linear. In this case, I've chosen something which is linear just to show you this gradient, because it's much easier to visualize and quantitate. But actually, you do see it almost reminds me of like running water when you put a drop of water and it's you have these splashes, which sort of strands of water that go. It's a little like that, except they grow and retract. The important thing is, how this is supposed to grow is the following. You take a vesicular lysosome, and then a vesicular lysosome at some point on this side, let me see if I can get, the players set up a calcium gradient. There are some channels which set up a calcium gradient. This recruits motors like kinesin on one side, dynein on the other side, and then both of them sit on a microtubule and pull the lysosome. Right? And so that's how a lysosome tubulates. But the important thing is, from this tubule, at some point, vesicles actually break off from the ends of the tubule, so that you have vesicles which have extremely high acidity or extremely high calcium. So the answer is that, you know, the tubules are not always focus straight. They can be sort of, sometimes they'll even be branched. So yeah.
I see. Okay, another question is that you didn't talk about your startup, this ESYA, ESYA. So if you could tell us about in 2 minutes or maybe in 1 minute, I mean, how actually it's useful to the society, how we can actually use, you know, especially from an enzyme point of view.
Yes, actually, I have now 2 startups thanks to macrophage, this tumor attenuation. So clearly, if you can send DNA devices to a particular place, one is you can use it as probes and make diagnostics. The other is you can use it for therapeutics. Therapeutics is what I spoke about right at the end, and that's been spun out into a company called Macrologic. ESYA is a company that deals with neurodegenerative diseases, where one of the things that we have, that we are now, it's becoming clear, is that neurodegenerative diseases are directly connected to dysfunctional lysosomes. And we have a way by which we can tell lysosomes are dysfunctional in live cells. So we can basically take your skin biopsy or blood-derived cells, look at the state of the lysosome, and this is an echo of what is happening in the brain. It can tell you how well your lysosomes are functioning, and your cells are still alive. So you can actually, if the signatures are, the ionic signatures are different, it's a sign that neurodegenerative disease has started setting in before actually the damage has occurred, right? Right now, they will diagnose you with Alzheimer's, or they'll diagnose you with Parkinson's. And by that time, it's too late. And these things you can't reverse. And so if we can catch lysosomal dysfunction very early, then you can put people on therapies early, and you can see what therapeutic is suitable for you. Right now, if you have Alzheimer's and you go to a doctor, they will prescribe you one of the 5 drugs, right? Then it's a crapshoot. Like, you don't know whether it's going to work for you or not. But now, if I have your cells, we can add 5 drugs to them, 5 different test tubes,
and see
which one your cells respond to. Put you on the drug that where your cells have shown the best reaction, and then you don't leave it to chance. So this is what the diagnostics company is about. And hopefully, if that works for Alzheimer's, then we have a slew of other neurodegenerative diseases that we can apply this to.
Okay, great. Yeah, thank you.
Thank you.
Okay, I think Shudipto and then Maitreyi. Shudipto?
Shudipto is on the phone, I think. Maybe Maitreyi.
Okay, Maitreyi.
Hi, Yamuna. Quick question. Very nice talk. So can you, maybe you mentioned it, but how do you propose that this Tudor device actually changes the morphology of the lysosome? Are you implicating the same set of interactions that you have on the top of the slide for MMP9? Or
yes, that's a great question. So let me tell you how this works, right? What Bhavya showed was that the moment it binds Ku basically, this sets off, it activates MMP. It has a domain that binds and activates matrix metalloprotease 9. And MMP9 acts through a receptor tyrosine kinase on the cell surface. We're not sure what it is, but bioinformatics and everything tells us that it may be something called protease activated receptor. We haven't yet done that. But this then activates an enzyme called PI3 kinase. So PI3 kinase, as you know, will put a lot of PI345P3, which is a lipid that is a very floppy, makes the cell membrane very, very floppy. And this is very important for a macrophage to actually, the membrane to remodel and then go and engulf, right? So this activates AKT, AKT activates mTOR. This is a very well-known cascade that activates mTOR. Now, the moment mTOR gets activated, what it does is that there are calcium channels, it will fall off. There are calcium channels that get activated as a result. And this sets up that gradient, right? So so for example, one of the proteins that it will recruit at this end is, let's say, AL8B, right? And AL8B then recruits SCIP, which then recruits kinesin. So downstream of mTOR falling off, all of this is actually very well known. In fact, when people have shown this entire pathway can also be activated by TLR4. So if I add lipopolysaccharide to the cell, which is a characteristic of inflammation, LPS can also activate PI3 kinase through TLR4. What Bhavya found was an alternative way by which PI3 kinase is activated without touching TLR4. So this is now a resting cell that is not inflamed at all. It's not inflamed. It's not autophagic. It's not a starving cell. This resting cell we've activated this pathway independent of TLR4, independent of autophagy sensing. And that's what allowed us to look at the role of tubular lysosomes in a resting cell. And just ask, when you don't have this complicated transcription of autophagy transcription program or inflammation transcription program, all of which impact the lysosome, right? So if you are otherwise looking at tubular lysosomes, you're always looking at it in the background of something which affects lysosome function, right? Lots of other players are upregulated. So what is the fundamental difference between a tubular lysosome and a vesicular lysosome was not known. But having found this pathway, we could actually ask that question. So but the pathway by which the lysosome itself gets tubulated is the canonical pathway. Thank you.
Okay. We will take student questions. Yes, Sandhya. Diksha, please unmute yourself.
Oh, yeah. Hello, Professor Yamuna. Really nice work. I wanted to ask if these RNA molecules can also form the duplex kind of structure based on cytosine-cytosine interaction that DNA molecules form.
It's a very good question. So, so RNA has this 2' hydroxyl, right? And actually what I didn't get into actually was, let me see if I can find it. Yeah. Share. Sorry. Share. Play this. Yeah. So RNA. I don't know. I don't know why this is. Yeah. 1 second. Give me a minute. Yep. So if you take a look at an eye motif, right? What we found is it's not like a box, okay? It's actually like a bar of chocolate. So the duplexes are actually intercalated, not in 90 degrees, but in sort of this very narrow 30 or 45 degrees angle. What that does is you have one very narrow groove here, right? Your two backbones are very, very close to each other. Other two backbones are very close to each other. So in DNA, you have this hydrogen bonded network between the phosphodiester's on the sugar, right? And that additional hydrogen bonding is what makes the system tetramerize. If you take RNA, RNA has this 2' hydroxyl. So as a result, it's got a stick poking out like that. So when these two strands come together, these sticks basically cause a steric hindrance. So RNA eye motifs are extremely unstable. However, you can get an RNA-DNA hybrid eye motif where you have one RNA strand, one DNA strand close to each other like this. And it can tolerate that, right? And so, yes, RNA can form I-motifs, but they're extremely unstable. You have to go to very low temperatures.
Yeah, okay. Thank you.
Okay, and now Arpan.
Yeah, hello, ma'am. Very nice talk. Actually, I think the question has already been answered. It has been asked previously. But having that thing, so can I just ask an extended part of that question?
Yes.
Yeah. So you said that this tutor is actually responsible for making this elongated lysosomes. But then also you stated that there are this budding of these spherical lysosomes from the terminal end of these tubular lysosomes. So does that actually result in a heterogeneity which is very difficult to understand in case of different sets of conditions when these cells are subjected to?
Yeah, beautiful question. So I sometimes wonder, this is full speculation, okay? So here you have this very nice thing where you have high calcium, low acidity, low calcium, high acidity. What people have seen is when you inflame a cell, let's say I add LPS or it's encountered a bacterium or whatever, the immune cell goes crazy, right? It will start tubulating its lysosomes, and its cell is like the membrane surface is ruffling. You can, if you see an activated cell, it's activated. You don't need to even map your lysosomes. Okay, so now under these conditions, your lysosomes are tubulating and going on, and then you need to have methods by which I can fuse and release molecules to the cell surface like MMP9, right? So I have to have high calcium in those vesicles, right? So those calcium is what enables fusion. So one end, which is pointing towards the cell periphery, if it breaks off into a little vesicle, that's a way by which I can fuse and send and throw out things like MMP9 onto the cell surface. The other end, it breaks off. You have a high acidity vesicle. These high vesicular lysosomes, what people have seen when they add LPS, they say proteolytic activity goes up, they'll say, right? When they've done, and they're looking at overall proteolytic activity in lysosomes. But we know that tubular lysosomes, proteolytic activity is less. Actually, what we showed was in vesicular lysosomes, proteolytic activity, when you add LPS, you have these high acidity lysosomes and proteolytic activity is super high, right? So now you can see that these tubular lysosomes are doing two things. Here, go fuse and make the membrane go nuts. And then here, start degrading, hyperactivate degrading and degrade the pathogen. So there are these two things that are possible. The other possibility is that, your phagosome, which has been formed, that phagosome has to fuse with the lysosome for a lot of things to be delivered. So this tubular lysosome, the more number of tubular lysosomes we found forms, the more number of phagosome lysosome contacts you have. So this is a way by which the system is able to generate and fuse and deliver a whole load of lysosomal enzymes to the phagosome, is what we think. And yes, lysosomes are highly heterogeneous. Biologists, if you look at it, they look at lysosomes, and if it has LAMP1, then it's a lysosome. If it doesn't have LAMP1, it's not a lysosome. So this is a digital way of describing a lysosome, right? But what I've just shown you is that you have so many populations. So if you now make chemical maps, you can chemically resolve lysosomes into different populations, which have they have LAMP, they have everything, yet the milieu is quite different. And this is quite interesting because lysosomes are very multifunctional organelles, right? They are involved in plasma membrane repair, they're involved in autophagy, they're involved in fusion, normal endocytic trafficking, they're involved in phagosome lysosome fusion. So it's not like Vishnu doing, multiple things. You can think of multiple lysosomes, each doing a different function. There was a time when people thought that all T cells are the same in 1974. Then there was a conference in 1974, and then they said, okay, then there's CD4 positive T cells, CD8 positive T cells. Now you had chemical markers, which then separated T cells into populations. Now you find this population has this function, that population is regulatory, this population is killer T cells, etc. So I believe we may be able to do something like that for lysosomes and start differentiating their function. That's a very interesting question. I don't think I answered.
Thank you so much. Yeah, I got it.
Thank you, Yamuna. Thank you, Arpan. Amal, could you unmute yourself?
Yeah. Hi, Yamuna. So my question is also regarding tubulated lysosomes. I mean, specifically, when you tubulate the lysosomes, do you know whether there is a segregation of lysosomal proteins into, various domains, and does that correlate with the calcium gradients that you see in that lines?
Oh my God. Yeah. No, this is a very, very interesting question. We don't know this, right?
Yeah.
I will tell you why. Because tubular lysosomes are really hard to do immunofluorescence. You have to fix them. The moment you fix the tubular lysosome, what happens is that you lose the calcium gradient, you lose the pH gradient. And if you don't have this gradient, the lysosome collapses. Nevertheless, Bhavya managed it. Few tubular lysosomes were still preserved. But the key was there weren't good enough antibodies to specific proteins on the lysosome that were interesting to probe. And although now we have a way to fix tubular lysosomes, the antibodies are not good enough or specific enough to probe selectively those channels or those proteins on the lysosome. However, if you look at what they had proposed, right? They had proposed that you have to have P2X4 channels on one side and TRPML channels on the other side. And that these were important to set up that gradient, that we also had to have VATPS on one side bringing in protons. So this was the entire, all the players that were known. And they postulated that these would have to be on one side, these guys would have to be on another side. And if that's the case, then there should be a gradient. We saw a gradient, right? We didn't expect it. And when Bhavya came to me saying that she found this gradient, I was like, no, there's some imaging artifact. No, don't worry about it. Because we were not aware of this prediction. And then she went off, did a whole load of beautiful experiments that showed us that that gradient is real. Then we went back and read a little more and realized this gradient has been predicted. Then we became very excited. So because it means that you might actually have this asymmetric distribution of these channels at least. I don't know about internal proteins, whether they would be sorted, but I think these are open questions.
Yeah, thanks, Amina.
Thank you, Amal. Are there any further questions, comments? I'm sure Yamuna would love. Okay, well, if there aren't any, I would like to thank, maybe the YouTube. No, YouTube doesn't have any questions, Sandhya. Okay, I would like to thank Yamuna once again for a fantastic talk and also giving a glimpse of what we can do with the kinds of tools that she's building up. You can sort of even decipher newer mechanisms that we were not aware of. So especially I love the fact that you have these two different shapes of these vesicular stuff. So now of course it cannot be vesicular, the columnar or yeah, great. Thank you, Yamuna, and I also thank many in the audience for sticking by till 7:30 in the evening and joining this colloquium. And also I would like to remind all of you that next week we have Professor Gagandeep Kang, who would be talking about vaccination and COVID responses. And this would be again an open colloquium at 4 p.m. Next week. And I would like to also thank our AG66 crew who stayed with us from 6 p.m. to 7:30 and let the lecture theater beam Yamuna's talk. There are, of course, we keep this on for people who are interested in still sitting in the auditorium and listening instead of just the desktops they have, so the laptops they have. So thank you again, Yamuna. And just can we have a little bit of a audio, unmuting the audio and just Yamuna.
Very nice talk, Yamuna. Excellent talk.
Yamuna, great talk.
Thank you.
Thank you very much.
It's always wonderful to come home.
And we would love to have you over for lunch. It's the traditional colloquium lunch, which happens here. So maybe when travel becomes a possibility again. So
Yes, indeed.
thanks, Yamuna.
Indeed.
Okay, bye.
Bye,
Yamuna.
guys.
Bye.
Happy New Year and stay safe.
Happy New Year to you. Thank you.